Hi everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. If you're watching these videos, you know that we're in the coronavirus shutdown and that I've migrated my courses to an online format, and how I deliver the information is through these videos. If you stumbled across these videos in YouTube, please know this: your instructor may emphasize or focus on different areas of the material or include information that I may not cover, and you need to learn it the way that your instructor wants you to learn it. Because these videos are intended for students who are enrolled in my Human Anatomy and Physiology 1 course at Del Mar College, and I'm going to present the information the way that I want my students to learn it now. Sometimes I simplify it early on, and then later on we get a little bit more complex with the material. So make sure that if you're enrolled in someone else's course, you learn the material that they cover in the manner that they want you to learn it. These videos are for my students. Now, if you are in my class, we're going to be on page 14 of the note set, and we're going to be covering chapter 3, the cellular level of organization. The first video we went over the levels of organization and some directional terminology and some language of anatomy. Also in lab, we're covering the body regions and all of that. The last couple of videos have been over the chemistry of the body, and only that little bit of chemistry that I want you to know for now, in a rather simplified fashion, and we'll get more complex with the material. So now, we're going to be talking about the cellular level of organization. If you look on page 14, what you will see is a list of organelles. There are a few additional organelles that are talked about in the textbook and the lab manual and listed in other places. I'm not going to cover those. I'm going to cover these. I'll mention them, but these are the ones that I want you to know for now, for this Human A&P class, and then later on we'll add in additional stuff as we need to, okay? So, when we start talking about these organelles, one of the things that we need to know is we're talking about what we call cytology. And if you watch the original videos, you know that cytology is the study of cells, cell structure, cell function, cell products, cell anatomy, cell physiology, cell morphology. Anything that says study of cells and cell blah, blah, blah is cytology, okay? Now, when we do cytology, we have to do all the organelles. We have to study the organelles. As we go through the levels of organization, we learn that atoms make up chemicals and molecules. Chemicals and molecules make up organelles. And then organelles make up cells. Cells make up tissues. Tissues make up organs, organ systems, and organisms. We went through the levels of organization. And one key principle that's important in anatomy and physiology is none of the levels of organization are more important than any other. They are all dependent upon each other. And so you should learn this concept. All levels of organization are interdependent, or we can say all levels of organization are dependent upon each other. A simple example is this: if one molecule in your body is not synthesized correctly, for example, insulin, a simple protein, if it's not synthesized properly, or if the organelles that are making that protein aren't functioning properly, then you cannot produce insulin. Insulin is necessary for our cells to take in sugars, and as you saw before, sugars are needed for energy. It's kind of like gasoline in your car. Without sugar and oxygen, your cells cannot make enough energy to perform their functions and will come crashing down and die. When a cell dies, a tissue dies. The tissue that is made up of that cell will start to become disrupted or enter the disease state or pathology. If the tissue dies, the organ made up of that tissue dies. The organ system that consists of that organ and others will come crashing down, and then the whole organism can come crashing down and die. So all the levels of organization are equally important. Now, we have to go over the organelle level in order to understand how cells function. Initially, I'm going to go through this list. I'm going to give you a description and a function for each organelle. It's going to be a little bit monotonous or boring because we're just going to write some words down and kind of, like memorizing the dictionary. But I'm going to put it all together in the end, either in this video or an additional video, because I'm trying to keep them about 35 minutes each. And we'll talk about how the cell functions as a whole, with all of these organelles being major players. So bear with me, okay? The first organelle listed is called the cell membrane. Now, when we talk about the cell membrane, one of the things we need to do is we need to describe it. The simplest description of the cell membrane is it's a lipid bilayer. What that means is it's 2 layers of lipids. And there's a reason for that. We're going to see this in the future when we talk about cellular transport and the cell membrane in great detail. And it's not just a lipid bilayer, it's very complex with some proteins and carbohydrates and cholesterol mixed in. But for now, we're just going to call it a lipid bilayer. And one of the things that we can talk about it we can say about it is this. Its functions are, it does isolation, protection, controls permeability, and provides sensitivity. Those are the four main functions of the cell membrane. It isolates the inside of the cell from the outside world. So if there are harmful substances and things in the outside world that could do damage to the cell, they cannot penetrate through the cell membrane, hopefully. So it isolates the internal environment or the intracellular environment from the extracellular environment. So therefore it protects the inside of the cell and all the organelles inside the cell from harmful chemicals and substances and things. It controls permeability. To permeate means to pass through. If the cell were freely permeable, then anything could pass into and out of the cell. We don't want valuable proteins, and certain chemicals and substances and organelles to just exit the cell. That would be like your heart literally jumping out of your chest or your stomach falling out of your body. You couldn't survive that way. So it controls what enters and exits the cell. Anything that enters or exits the cell must pass through this lipid bilayer in some way. And so we're going to discuss that in great detail. The last function that we can say for the cell membrane is it provides sensitivity. Our cells have the ability to sense certain chemicals and substances and boundaries surrounding them. One of the things that's very interesting is that in some cells, if you grow them in a petri dish on what's called agar, some of these cells, as long as you feed them, will start to divide until they cover the entire surface. And when there's no more room to grow, they'll stop growing. The only way they could do that is if they know that they've reached the limit of the space, the capacity of the space inside that area. Not all cells do that, but some do. Cells have the ability to sense specific chemicals and substances in the external environment, therefore taking some things in and holding some things out and sensing the presence of other cells around them sometimes. So now, before I move on, I always learned it this way. It's a simple way to learn stuff, to memorize for an exam. I learned it as IPS, or one of my classmates said PIPS, which is isolation, protection, permeability, and sensitivity. So when we ask a question, which of the following would be the functions of the cell membrane, you can come up with those four things pretty easily. Isolation, protection, permeability, sensitivity. Or protection and isolation, permeability, sensitivity. Okay? You got to come up with ways to learn this stuff and make it stick to your brain for the long haul for exams. I'll share with you a few of those tips and tricks. Now the second organelle that's listed on our list of things to know is called the cytoplasm. And the way that I define this is the cytoplasm is the entire cellular contents. It's everything inside the cell. And I'm going to switch markers because this one's getting a little bit weak, but there are two major components to the cytoplasm. There's a stuff called the cytosol, plus the additional organelles. So, that begs the question, what is the cytosol? Well, you should know cyto means cell, and the sol part comes from the word solution. So we tend to think of the cytosol as the fluid part of the cell. And then the organelles would be all the substances floating in that fluid. If you think about it, think about a little bag of pond water. If I could take a water balloon and fill it up with some pond water, that would mimic what a cell would be. It's a cell membrane, that would be the balloon, and the fluid inside would be the cytosol. Now, if I could take a bunch of stuff and have it floating and suspended in there, so that when we look in there, we see things floating around and moving around inside of there, all the stuff suspended in the cytosol would be the other organelles that we're about to discuss, okay? So what you need to know about the cytoplasm is it's everything inside the cell membrane. And they call it cytoplasm because it's not water. it is a watery environment that has lots of sugars and proteins and other substances mixed in and some organelles, so it gets rather thick and viscous, almost like syrup. So imagine if I took a water balloon, filled it with syrup, and then dropped a bunch of candies inside of it that were floating inside there. The candies would be the organelles, the syrup would be the cytosol, and then all of that together would be called cytoplasm because it's rather plasma-like or thick. Now the next organelle we can talk about is going to be the cytoskeleton. And I mentioned this in one of the previous videos. When we talk about the cytoskeleton, cyto means cell, and skeleton means exactly that. But the cytoskeleton is a network of, man, I'm going to have to keep changing markers until I find one that works really good, of interconnected proteins. It provides shape and support for the cell. So, one of the things that we do know, sorry, I wanted to make sure that everything's within my frame because it's just me and a cell phone. I always think of the cytoskeleton as like the tent poles inside of a tent. And if you rearrange the tent poles, the tent would change shape. And what holds the tent up is the tent poles. Well, the cytoskeleton is this network of interconnected proteins that are like little rod-like proteins. And they give the cell its shape and support. Some of the cells are rather amorphous. Some cells are rather cube-shaped. Some are long and slender cells. And the nucleus is I always draw the nucleus inside the cell. The shape of the cell, some cells are going to be rather star-shaped or these funky shapes. The shape of the cell is dependent upon the arrangement of the cytoskeleton, which is simply nothing more than some structural proteins that give it its shape and support. One of the things that the cytoskeleton can do is it provides an avenue for cellular transport, and that's a really interesting topic, but I don't want to go down that road right now. We'll talk about that in due time. So that's the main function of the cytoskeleton, kind of like your own skeleton. Now the next organelle we want to talk about is going to be the cilia and the microvilli. So, cilia. These are Mr. Long definitions, but it's the way that I want you to learn it. If you look them up in the book, it might be slightly different, but at least start here, then go read the chapter. Never substitute reading the chapter for just watching the videos. You have to read the chapter and learn the technical language as well. Cilia are long membranous extensions that move substances along the surface of the cell. They're long membrane extensions that move substances along the surface of the cell. So if I'm a cell, my arms would be like cilia flapping. And the reason they move is because the cytoskeleton in there can be moved and cause the cell to move around like this. Okay. Now, when I look at a typical cell, let's say I have a cell that looks like this. It does not have cilia. And then I have another cell that looks like this. These long membrane extensions would be cilia. And we find cilia on cells. Not all cells have cilia, only a few do. But we find them on the cells where we don't want stuff to stick around a lot of times. So if some debris or something bumps into the cell here, as the cilia are flapping around, they can push this substance all along the surface of the cell and move it to another area of the body where we can get rid of it. So they're long membrane extensions that move substances along the surface of the cell. Okay? That's what you need to know about cilia. Now I'm going to erase all this. I'm going to leave this little drawing here because I'm going to talk about microvilli. And microvilli share somewhat of a similar description. So, microvilli. Villi comes from the word villus, which means finger, and micro means tiny. So we sometimes describe these as short membranous extensions, that increase the surface area of a cell for absorption. And this is the key word. When we find areas of the body where we want to do a great deal of absorbing from the surrounding area, then cells will have these very short membrane extensions. And whenever a substance bumps into them, they can take that substance into the cell. So we find cilia where we want to keep stuff moving along. We find microvilli where we want to absorb things. I often think of cilia as the arms at a concert. If you see a crowd, it's like all the cells and all their arms are like cilia. If someone jumps off a stage, you can push them around and move them back to the stage. That would be like cilia. Microvilli are going to grab things and pull them in. So they're little finger-like structures or short membranous extensions that increase the surface area for absorption. I want to show you one other thing, okay? If I were to look down on the top of this cell, if I could stand up and look down on top of it, and let's say the surface area of the top of this cell is about the size of this sheet of paper. It's 8.5 by 11, but for mathematics, to make it simpler, let's just say it's 8 by 11. That's 88 square inches. So that if any food or nutrients or anything are passing by here, I got 88 square inches in which to absorb it before it moves on. And your digestive tract is where we find a lot of cells that have microvilli for absorption. Well, what if I take this exact same area, okay? Now watch. If I were to try to outline this piece of paper here, that's roughly my 88 square inches. This little square. Now watch this. This is pretty cool. Mother Nature is beautiful in her design. And if I take this exact same sheet of paper and I fold it up. So some people describe microvilli as membranous folds or membranous extensions. But now look, I've taken the same 88 square inches and I can cram it in here and I can fit 3 of these in the same amount of space, tripling my surface area. And the reason this is important is because when we are lucky enough to eat, because sadly enough, most of the world's population doesn't know where their next meal is coming from. So if you are lucky enough to get some nutrition, then before it comes out the other end and you lose it, you want maximum surface area to absorb as many nutrients as possible. And along the surface of the cell, there's some special proteins and transport molecules that are moving stuff in. The more surface area, the more transporters that I can have to pull the nutrients in. So now, we've covered those structures. Let's move on to a few others, okay? I'm going to finish this list up and do the cell story, and then we'll move on to other topics, okay? So now, one of the ones that's on your list is called centrioles. And centrioles may not make a whole lot of sense until I cover them in lab and we talk about mitosis in the laboratory. But the function of centrioles, and again, I'm going to keep changing markers till I find one that writes dark enough for you to see. Centrioles, their main function is that they separate, oops, I can't spell right now. What they do is they are going to separate chromosomes during cell division. And when we talk about cell division, it's a special process that we're going to call mitosis. We will also eventually talk about meiosis, another type of cell division. Sorry, I keep dropping markers. But for now, you need to know this. There are these structures inside the cell called chromosomes. And at some point, we're going to have to separate them out to either end of the cell. And the cell is going to pinch into half, forming 2 new cells that have the same chromosome makeup or the same genetic makeup. They're going to have, each one's going to have a copy of the same chromosomes. The process of separating and moving those chromosomes is performed by these little protein-like structures called centrioles. And that process of the cell dividing is called mitosis. So you need to know that centrioles are little protein-like structures that separate chromosomes during cell division. Something that we know about centrioles, by the way, is that the cell always has two of them, and under a microscope, when they're not functioning, they look like little tubules. To me, they almost look like little pieces of the Twizzlers candy that someone bit off and is holding. And they're always at right angles to each other, okay? They're rather tubular little groups of proteins sitting at right angles like this, no matter how you look at the cell and rotate it. So sometimes we describe them as the cell having two of them at right angles to each other. Now there's another definition here called the centrosome. And I don't know if you can see that marker very well. I'm having trouble with markers today, so I need to check them all and redo them. The centrosome is an area of the cell near the nucleus, usually, where centrioles are found. So they always make a big deal about the centrosome in book, but I don't really make a big deal about it. I'm more interested in the centrioles themselves. Two small tubular proteins that separate chromosomes during cell division. Okay? So now, we can erase the centrosome. We've talked about it. I'm not interested in it. All right? Now the next structure we're going to talk about is going to be ribosomes. And one of the things I want y'all to know is that all ribosomes do the exact same thing. So when I talk about ribosomes, I'm going to give you an extra definition of this because I'm not going to talk about free or fixed. I'm going to talk about all ribosomes first. The function of all ribosomes is that they synthesize proteins. Or one of the things we can say, another way to say that is that they build proteins from amino acids. So, I told you in one of the lectures that proteins are long chains of amino acids, also called polypeptides. And if these markers represent amino acids, as I start to assemble them together in a specific order, I would be a ribosome. And I would be assembling the amino acids in a specific order. And then when I have all the amino acids assembled, what I have now is a protein. So I have just synthesized or built a protein out of amino acids. I'm the ribosome. That's what all ribosomes do. Now, the way they know which amino acids to put in what order is they get some instructions from the nucleus called messenger RNA. And the messenger RNA is literally like me getting a message and reading it that says, please put this amino acid first, this amino acid goes second, this amino acid goes third. So the messenger RNA is literally the assembly instructions on how to put a protein together. The organelle that assembles the protein or synthesizes it is a ribosome. Now, there are two different categories of ribosomes. So, what I want to do is distinguish between them for you so that you understand the difference between them. Okay. There are free ribosomes. If I were to describe free ribosomes, we would say that they are floating freely in the cytoplasm. Now this isn't going to make a whole lot of sense until I talk about the next one, but they are floating freely in the cytoplasm. And what I'm going to tell you is sort of a generalization. It's not 100% accurate all the time, but for now let's keep it simple and then we'll add difficulty and complexity later on. So to keep it simple for now, one of the things we know about free ribosomes is that they are going to synthesize proteins that remain in the cell. Now again, that's not going to make a huge amount of a difference for you or explain a lot for you until I talk about bound ribosomes or what we call fixed ribosomes. So when we talk about fixed ribosomes, and some people refer to them as bound ribosomes, we say that they are attached to the rough ER, and the ER stands for Endoplasmic Reticulum. We're going to cover that organelle in just a moment. But the reason they're called fixed or bound ribosomes is that they are fixed to or stuck to or bound to another organelle called the endoplasmic reticulum. And because all these little organelles are stuck to it, it looks rather rough or bumpy. There's also a smooth endoplasmic reticulum. We'll talk about that in a moment. So one of the main differences between free and fixed ribosomes is that free ribosomes are floating around inside the cell. Bound ribosomes or fixed ribosomes are not floating freely. They're attached to another organelle and kind of stuck to it. Now, one of the things that a lot of textbooks can indicate is that free ribosomes are always free and bound ribosomes are always bound. And that is not true. When the ribosome receives the instructions called messenger RNA, and those instructions come to the ribosome, it not only will tell it how to put the amino acids in the correct order in which to assemble it, it'll also say, hey, by the way, this protein is going to be secreted outside the cell, or this protein is going to remain inside the cell. If the protein remains inside the cell, when the ribosome assembles it and puts all the amino acids together, when it's done, it will release the protein off into the cell to go do its function. And it will release the message and then look for more instructions to build another protein. So, free ribosomes receive messenger RNA, read the instructions, assemble the protein, and release the protein in the cytoplasm, and most of the time that protein is going to stay somewhere inside the cytoplasm and do some kind of function inside the cell or be part of the cytoskeleton and other things. For bound ribosomes, when they get the instructions, sorry, I have to keep bending over, I keep dropping things, sometimes on purpose. When they start to assemble the protein out of all these amino acids, the instructions also tell them, in some unique chemical language, that this protein is going to be secreted outside of the cell. If the protein is going to be secreted outside of the cell, excuse me for a second, then we have this long tubular structure called the endoplasmic reticulum. And when the ribosome gets the instructions, it will bind to, this was originally a free ribosome that becomes bound to the endoplasmic reticulum, and as it starts building the protein, it literally shoves that protein into the endoplasmic reticulum. When it's done, it can release the message, and it can go float freely again and become a free ribosome. So essentially, you need to know this. All ribosomes synthesize or build proteins out of amino acids, and they use some instructions called messenger RNA. If the protein is going to stay inside the cell, it is usually synthesized on a free ribosome that's floating freely in the cytoplasm. So it's going to synthesize proteins that remain in the cell. If the protein is going to be transported in the cell and either stuck in a cell membrane or secreted, then it's going to be made by a fixed or bound ribosome. So we can say the function of bound ribosomes is that they synthesize proteins that are secreted from the cell. They're going to be released by one cell into the extracellular fluid because another cell might need that protein and may not be able to make it. Okay? So, now this is an oversimplified view of this. If I were teaching, a junior or senior level cell biology course or molecular biology course like I used to do, I would go into much more detail. But for now, there's so much shotgun spray of information, we're going to keep it simple. Know this about free and fixed ribosomes. Got it? Good. Now, let me erase this. Let me talk about the endoplasmic reticulum, the Golgi apparatus, and mitochondria and lysosomes. Okay? So, when it comes to, there is a structure called the nucleolus. I'm not going to worry a lot about the nucleolus. I'll talk about it. We do know that about the nucleus has a double membrane. With pores. And that is called the nuclear envelope. The nuclear membrane is very often referred to as the nuclear envelope. And what it is it's two lipid bilayers. It's very similar to the cell membrane, only there's a second layer of it. And if we were to look at it in 3D, what we would see is that in some areas, the nuclear membrane has little gaps in it. If I were to try to draw this sort of in 3 dimension, there's these little holes or openings, like this. Those pores or openings, called nuclear pores, allow stuff to enter or exit or permeate the nucleus. So sometimes a messenger RNA needs to be released from the nucleus out into the cytoplasm, and then a ribosome can read the messenger RNA and assemble a protein from it. So the nucleus has permeability, and the permeability comes through these little openings called nuclear pores. They're very complex structures, and we're not going to get into that now. But when we describe the nucleus, we usually say that it has a double membrane that has pores in it called the nuclear envelope. And one of the things that's found inside the nucleus that I want you to know about is that it houses, or some people say stores and protects, the genetic material of the cell. And this is really not the best description, but that's where all the DNA is found inside the cell. Deoxyribonucleic acid, the genetic material. The genetic material, really, the DNA is the instructions on how to assemble every protein that this cell is capable of making. So imagine if I had instructions on how to make glass jewelry out of little glass beads. You could make necklaces and earrings and bracelets and hippie curtains or whatever. All of these glass beaded type structures. If we had a factory that made those, I would have to have the instructions for every product that we're capable of making. And if someone was going to have a Mardi Gras parade and order a bunch of different things, then we would have to pump out thousands of these products. The worker out in the factory producing them would be a ribosome. How each ribosome knows which protein to make is it gets some instructions called messenger RNA. And that messenger RNA is just a little single set of instructions for one protein for that particular ribosome. But inside the nucleus, I have these long double helical molecules called DNA, and along these molecules, there are areas that are the instructions for different proteins. Maybe protein 1, 2, and 3 on this chromosome or piece of DNA, and on this one I can have even more proteins sometimes. So each piece of DNA, each chromosome inside the nucleus has the instructions on how to assemble all these different proteins. And the way the cell knows how to assemble a specific protein at a specific time is there's some chemical or molecular signal that tells it to do that. I can turn on a gene, rewrite that gene into messenger RNA, send the mRNA out to the ribosome, and I can build that protein from those instructions. And the ribosome doesn't have to come in here and search all the different genes. It just gets the instructions that say, build this protein now. So imagine if we had a factory and I had a giant book on how to assemble every one of our glass bead products. The guys out in the factory, the guys and gals working out in the factory don't need to read all those. If I have this half of the room making one specific protein, then I can take those instructions, make hundreds of copies, and give them to those students, and then they can start assembling the proteins. And if I have a different set for this group, then I can go into a different gene, rewrite the instructions into hundreds of copies, send them out to the ribosomes, and they build all these different proteins. This is how the cell can make thousands of proteins in a short period of time. So, the nucleus is a double membrane with pores called the nuclear envelope that houses, stores, and protects the genetic material of the cell, or it stores the DNA in the chromosomes. Okay? Now, that's a simplified view, but we're going to get more complex over time. I have a few more organelles to go over, so let's wrap this up. I'll probably close out this video, and I'll just do another video of the cell story. I'm kind of making this up as I go along because of the circumstances. Now, one of the organelles that we haven't discussed is the rough and the smooth endoplasmic reticulum. The reason I glossed over the nucleolus is those are little dense areas on the nucleus where ribosomes are assembled, and I don't really care about that. If this were cell and molecular biology, we'd talk about the nucleolus. When it comes to the endoplasmic reticulum, there's one called the rough endoplasmic reticulum. And sometimes we say RER for rough ER. Now, you guys need to know that when we're learning this stuff in lab, you're going to have to spell the words out. You cannot abbreviate. But the rough endoplasmic reticulum is this. This is how, to me, this is the best way to describe it. It is a network of interconnected membranous tubules with ribosomes attached. So it's a network of interconnected membranous tubules. So imagine if I have a tube of membrane that snakes around like this. If I took another tube of membrane over here and I let it snake around and I drew a third one, I would have all these membranous tubules. Well, it turns out that sometimes they are connected to each other so that I have a series of interconnected membranous tubules, and they can be interconnected in a number of places so that anything that's flowing inside this membrane can move around over here or make it over here and flow around in this maze of membranes. So it's a network of interconnected membranous tubules. What makes it look rough is that on the surface of it, there's all these little bumps. When we look under an electron microscope, scientists saw all these little bumps all over the surface of part of the endoplasmic reticulum. And because it looked rather rough or bumpy on the surface, they called it rough endoplasmic reticulum. It turns out that those little bumps are simply bound ribosomes. And as these ribosomes might be floating out here, when the ribosome gets a message of mRNA, I said just a few minutes ago that that message may say, hey, this protein is going to be secreted. Before the ribosome starts synthesizing the protein, or as it begins synthesizing it, it can go and attach. And as it reads the messenger RNA, it will build a protein and then shove that protein into the endoplasmic reticulum. And now that protein can float around and be transported all over inside the endoplasmic reticulum. So that's what rough endoplasmic reticulum looks like, and its function is that it's going to synthesize proteins that are going to be moved around in little transport vesicles. So if I were to write out the function of the rough endoplasmic reticulum, I would write it this way. It can synthesize, and quite honestly, the endoplasmic reticulum isn't synthesizing the protein, it's the ribosomes that make it rough. But it can synthesize proteins to be transported. I'm going to erase part of this. In the cell for secretion. Now, this is not 100% accurate in all instances, but for now, we're going to keep it simple, okay? So eventually, what will happen is, if I need to move a bunch of proteins around, I can stick those little proteins. Let's say I have a bunch of protein right here. And I have my ribosomes that are sitting on the surface of this, which synthesize that protein, okay? Let's say I'll put them over here. What can happen is this. One of the things we're going to learn when we talk about lipids is that lipids stick to other lipids, and they don't like to interact with water. Kind of like if you took 2 drops of oil and put them in some water, if they came together, they would join together and form a much bigger blob. So I can take 2 different lipids and they will form together into a larger bubble of lipids, or if I whip it with a whisk or a fork, I can break those little bubbles up into smaller ones. So lipids can fuse together, or they can pinch apart but still seal each other off. And what can happen over time is the rough endoplasmic reticulum, using some proteins, can start to pinch itself in, and eventually it will pinch off a little bubble of membrane that's going to be transporting proteins around inside the cell. Eventually, usually those proteins will be secreted. Okay? We call that process protein sorting. I don't know which proteins go where, but I'm not going to get into that. That's a really interesting topic I did some of my master's work on, but no well. Now, we've talked about the rough endoplasmic reticulum. Let's talk about the smooth endoplasmic reticulum. And I'm going to leave some of this information up because a lot of it is the same. The smooth endoplasmic reticulum is a network of interconnected membranous tubules with no ribosomes attached. So this part over here would be smooth ER because there's no ribosomes attached to it. This over here would be rough endoplasmic reticulum because it has ribosomes attached to it. It's the same endoplasmic reticulum, but if there's no ribosomes, it looks smooth on the surface. If there are ribosomes, it looks rather rough on the surface. The ribosomes synthesize proteins. We pinch off a bubble of membrane to transport those proteins. By the way, this little bubble of membrane that contains proteins is called a transport vesicle. And it would be filled with proteins that are going to be moved around inside the cell. They're called a transport vesicle. Vesicle means little vessel, little container. Now, what if the smooth ER pinches off a bubble of membrane? Because there's no ribosomes, there's no protein in there most likely, but inside this fluid called the endoplasm, that fluid does have some sugars in it and some lipids, and there's lipids in the membrane of the rough and smooth ER and in these vesicles. They're the same lipids that make up the cell membrane and the nuclear membrane. So since this is only going to be transporting a little bit of sugar and some lipids, very often we can say that the function of the smooth ER is this.